Hello, everybody. Thank you very much, organizer, for having me here. It's a pleasure. I belong to ITERN project, which is the Marie Curie Initial Training Network, aiming to develop an image tissue engineering constructs. The project unites 6 European countries and employs 12 PhD students and 1 postdoc. And we are working together, we are collaborating together and exchanging technologies. My role in the project as a chemist is to develop an imaging agents for magnetic resonance imaging, MRI, and design the conjugation methodologies to engineer tissues. So within my talk, I would like to share with you my latest achievements towards completing this goal. Tissue engineering has evolved as an interdisciplinary technology which combines the principles of life, materials, and engineering sciences in order to support, regenerate, and replace damaged tissues. The typical general concept of tissue engineering can be seen in this figure where the cells are isolated from the patient, recultivated in vitro, and seeded onto biomaterial. Biomaterial can be serves as a temporary scaffold, which not only provides a mechanical support, but also helps to reorganize the cells and forms a functional tissue. So together with cells are placed growth factors. They are very important components, but also small molecules and nanoparticles, which depends on application. Once the form scaffold is ready, it goes to bioreactor to form functional tissue, and then goes back to the patient to cure or replace the tissue or organ. There are a lot of, or I would say currently, there is a lot of potential observed for various classes of materials for potential clinical applications. One of them are hydrogels, because because of their internal structure, they excellently mimic soft tissues, but also the customized pastes, such as calcium phosphate cements, which are used for bone reconstructions, but also a lot of attention is given for fibrous biomaterials or foams, or sometimes the combinations. Within this talk, I would like to focus mainly on designing nanoparticles and small molecules to be conjugated to various biomaterials. As tissue engineering matures, the new technologies are required for biomaterials. And actually, therefore, to assess the very advanced applications, the engineers need to have a versatile method, imaging method, capable for visualization, monitoring not only a morphology, but also a function and molecular information, I would say. So general procedure to assess the behavior and function of the tissue engineering construct is presented in here, and it starts from designing of the material and its characterization towards its effectiveness. And this sometimes can take ages, 4 weeks, months, or sometimes a full PhD study. But once the material is developed and characterized, it can go beyond the lab and go to the preclinical tests on the animal models. And still, there are a lot of tissue engineering studies relying on ex vivo information, which is nothing wrong, but it is very limiting. For instance, histology, it's a type of ex vivo experiment which gives only one-time characterization point, and moreover, it doesn't give us information about the volume of the implant, as well as it's not possible to detect really early stage immune rejection. That's why magnetic resonance imaging, MRI, is a key. It's a key instrument in pharmaceutical and biomedical research. It can give us a lot more information, such as volume of the implant, 3D, three-dimensional shape and structure of the implant, and very importantly, it can gives us precise placement, anatomical placement of this implant in the body. But I do think personally that it's something very important from the chemical point of view. MRI can answer as a question towards understanding the biodegradation of the polymer in vivo. However, despite its a lot of positives, it has some drawbacks as well. So one of the drawbacks is the fact that imaging some of the classes of materials are quite challenging. For instance, hydrogel. I'm going to mainly focus on the materials which are in the key of my interest, so such as hydrogel, because they contain 90.9, sometimes, according to my hydrogels, they contain a lot of water. That's why, as you can see in here, there is basically no difference between the extracellular matrix and the hydrogel in here. As well as calcium phosphate cements for hard tissue reconstructions. Once you can see in here that it has been created a defect in the bone and filled with CPC cement and imaged by MRI. Unfortunately, there is no difference between these classes of materials because CPC has it's very dense material. It doesn't have enough of water to give a sufficient contrast enhancement by MRI. For this reason, application of paramagnetic metals for magnetic resonance imaging is a key solution to overcome the intrinsic material limitations. I have divided the contrast agents for 3 groups, which are based on, for instance, metal-ion complexes. These are, we call it DO3A ligands for gadolinium complexation. Currently, they are used in clinic. As well, we can use gadolinium oxide and iron oxide. So actually, this one is very known and popular in the science, nanoparticles, to enhance or T1 contrast or T2 contrast. Or also, we can more go further towards modern science right now, and the key interests actually right now, which are multimodal imaging agents. What is interesting in them that we can combine different modalities together and obtain even more information to understand the full chemistry and biology. So for instance, taking nanoparticles, which can be iron oxide or can be gadolinium oxide, and functionalize their surface by addition of various molecules, such as organic dyes for optical or radionuclides with different half-life times. We can have both PET MRI contrast agent, MRI optical, and so on. Right now, I would like to speak about the hydrogels used within my project for soft tissue reconstructions. The group from Netherlands, collaborators from Netherlands, have developed hydrogel, thermosensitive hydrogel, which is based on, that possesses a stiff and helical structure. And because of the fact that the polymer is grafted with ethylene glycol chain, they caused very specific hydrophobic effect, and this polymer as a construct showed tunable thermal transitions. As you can see in here, when the aqueous solution of the polymer is kept below 18 degrees Celsius, it is in a liquid state. But when, for instance, it is implanted in vivo, so in the room, room temperature and body temperatures conditions, it forms a gel. So in here, in atomic force analysis, microscope analysis, you can see the 3D structure of the polymer. But how I can, from the chemist's point of view, how I can conjugate MRI contrast agents to such as construct. So I asked the group to functionalize the hydrogel, and they have added azide groups onto one of the ethylene glycol chains, which using a click chemistry reaction approach, I would say clicked, so conjugate various MRI contrast agents. And why I chose click chemistry reaction, it is because it's quite fast, relatively fast, it's very selective, and most importantly, doesn't create any bioproduct, any toxic bioproduct, which is, of course, a super extra issue if we are considering biomedical applications of these ones. So I have chosen and synthesized DO3A derivatives for gadolinium complexation, functionalized by addition of different alkane chains in here. DO3A contains 7 coordination sites because, as we all know, gadolinium in a free metal ion form is very toxic in vivo. That's why making a complex with these ligands, it's basically make the whole construct safe for clinical use. And these constructs were characterized in terms of their contrast efficiency. And as you can see in this phantom image, they provide significant T1 contrast enhancements compared to water. And the highest relaxation rate, so the best contrast agent from out of these 3, revealed to be this construct in here. So before conjugating them to the hydrogels, of course, I had to optimize the conjugation conditions. Because of the very specific maybe not specific, it's because of the gel is very sensitive towards low pH or too high pH, towards various organic solvents. These 2 conditions, which I firstly tried to optimize, didn't work. So I had to turn towards the third option, when it was used copper in situ generation of copper-1 species in water-entered butanol. In order to monitor the kinetics of this conjugation, I have performed the model click chemistry reactions, a reaction using the small molecular benzyl azide, which hold the product I could observe and monitor by mass spectrometry. So this reaction was fully completed by detecting the main product and depletion of the starting materials by mass spectrometry. So knowing already the kinetics of this reaction, I have clicked 3 different alkyne terminated DO3A complexes onto the hydrogel backbone. And this is the triazyl ring, which basically confirms the conjugation efficiency, whether the conjugation was successful or not. In order to characterize these conjugates, as a chemist, I know that the golden standard to characterize any chemical compound is IR spectroscopy, because it's very fast and quick and can give already some crucial answers. So I perform a IR spectroscopy, and I could already detect the presence of the functional groups which are related to triazyl ring. So already I knew the answer that it worked. I performed a relaxivity study using clinical 3T scanner, and all of the constructs shown significant T1 signal improvement compared to water signal. Elemental analysis has shown that the excess of the microcycle, I added extra equivalent of the macrocycle for this conjugation to push it to completion, and all the excess of gadolinium has been removed by dialysis after 12 hours. Unfortunately, I need to say in here that I wasn't expecting that, but the copper, as a toxic catalyst, is trapped within the hydrogel matrix. So currently my master students are working on the successful efficient removal of copper-1 catalyst, because at this stage the hydrogel cannot be proceed to in vivo because of the toxic copper. So because of that, I had to develop a new strategy to overcome this limitation. So I synthesized similar DO3A complexes, but suitable for copper-free click chemistry reactions. So I synthesized this molecule in here, which vary by here the amine spacer link, where I have I was measuring the efficiency of the relaxation, and after clicking these molecules onto hydrogel, just the reaction happened in water, so without any additives, I could clearly see a significant improvement of the T1 signal intensity compared to unfunctionalized hydrogel and water. Water and hydrogel gives the same signal, so no signal. So in here you can see the improvement in phantoms. From basically no signal in unlabeled hydrogel, I could already see a bright signal enhancement. as well as because of the sensitivity and selectivity of this reaction, the yield of a conjugation is definitely much higher. So content of gadolinium in the hydrogel, it was as expected, is definitely higher compared to a copper-1 click chemistry reaction. This can be due to insufficient amount of catalytic centers which were available to push the reaction to completion. That's why probably it is that big difference. That was covalent attachment methodology. But I was also wondering if it's possible to label the gel in an efficient way, fast way, and without involving too much chemistry. So I wanted to investigate, so as I called, a non-covalent attachment methodology to hydrogel using nanoparticles. So I have synthesized iron oxide nanoparticles, which were coated with polyacrylic acid, varying with a molecular wave. So when I used a smaller molecular wave polyacrylic acid, so I'm going to call it caping agent, definitely difference in morphology I could observe, which translated also for hydrodynamic size measured in water. These nanoparticles are having a low polydispersity index, and they are suitable for in vivo application due to their quite low hydrodynamic size. Also, these two type of nanoparticles showed significant T2 signal improvement compared to water fountains, but also because of their ultra small sizes. The core size of these nanoparticles are about 5-6 nanometers. They show also significant T1 improvement compared to water phantoms. So, having these nanoparticles prepared, I wanted to encapsulate them into hydrogel matrix. But before I'm going to speak about that, I wanted to recap the AFM image to show you how big the pores are within this with these hydrogels. So imagine that if I would encapsulate the bare nanoparticles with the core size about 5 to 6 nanometers, within such a hydrogel, they would be simply washed away, wouldn't they? So I had to think, and before I made the synthesis, I already hypothesized that the residence times of these nanoparticles can be tuned playing with the molecular wave of caping agent. So the hypothesis of mine was, as you can see in here, that the longer the chain of the polymer is, they can work like a hook to the helical structure of the hydrogel. So to prove this concept, I did a release study of these nanoparticles in water, and as I predicted, the bigger the molecular wave of the polyacrylic acid is, the longer residence times is within the hydrogel. So, but I still wasn't that much satisfied because of the experimental conditions. I thought that maybe the nanoparticles are released due to the surface erosion of the biomaterials on the top of in here. So I performed, as I call it, mobility study. So I wanted to answer whether they are possible to migrate within the hydrogel, and then because of the migration, they are able to be attached to the hydrogel itself within the matrix. So the mobility study, as it is shown as an experimental procedure, has revealed that over the time, the T2 signal intensity of the gel is significantly increased. And why it's increased? Because of the presence of the nanoparticles that are coming in. So that proved that they can be washed out as the same way as they can go in. So there is specific interaction inside. And now I would like to turn towards calcium phosphate cements, which is I would call it the second chapter of my talk today. The group from Netherlands, the different group in Netherlands, has experience on developing CPC calcium phosphate cements. They already performed some preliminary study by incorporation of iron oxide nanoparticles into calcium phosphate cements, and they have published in 2013 their results. Well, it looks fantastic. They can obtain a really darkened signal coming from the implant side. However, as you can see in here, this flower-shaped dot doesn't correlate with a spherical size of the defect filled with cement. And why it is like that? Because the presence of the iron oxide caused blooming artifacts, which, as you can see in here, oversized the defect. So actually, this composition can't be translated into clinical because none of the clinicians wants to see oversized defect and basically bringing error to make a diagnosis. So I have decided to replace iron oxide over gadolinium oxide and to enhance T1 signal, but also decrease the image artifact. We hypothesized as well that the addition of the bisphosphonate groups onto nanoparticles can also enhance the new bone formation within a CPC once it is in vivo. So gadolinium oxide nanoparticles has been synthesized in a similar way that iron oxide. So it calls polyosynthesis approach using high boiling point alcohol. The presence of the pure phase gadolinium oxide was confirmed by XRD. And surface functionalization has been performed by addition of this type different various bisphosphonate, which differ by here the alkene chain. This one is a commercial available, and it calls alendronate. It is commonly used component for heart tissue reconstruction, and these were synthesized. So I have divided the gadolinium oxide nanoparticles into two groups to investigate their type of coating on the stability and the conjugation efficiency. So the first group has involved a coating, gadolinium oxide coated with polyacrylic acid with the low molecular weight. And the other group has been coated with silica, with the epoxide ring, which is very selective towards primary amine here. So this reaction has been performed under basic conditions in order to deprotonate the image and proceed the reaction to completion. Samples were characterized by elemental analysis. So in here, as you can see in here, there are no nitrogen before functionalization, and the drastic increase of the nitrogen content after functionalization. This gave me already a primary answer that the molecules has reacted with each other, and as well IR spectroscopy has been performed in order to confirm the successful functionalization. Relaxivity study was performed using clinical scanner 3T and preclinical scanner 11.7 Tesla in order to select the best candidates for future in vivo application. So based on this relaxivity curves, I could already see that the gadolinium oxide coated with silica gave significant contrast improvements, D1 contrast improvements compared to the ones which were coated with polyacrylic acid. So now was the question, which of the type of bisphosphonate really gave the best result? So for silica-coated gadolinium oxide, I have performed the same sort of study but using this time 11.7 Tesla because of the fact that future in vivo will be done in this type of scanners. And the longer the alkene chain in the bisphosphonate, so the longer the bigger the molecule is, the better relaxation I can get. So this candidate has been selected from out of this full library to be mixed with CPC cement, and further mechanical test is ongoing, as well as long as with the toxicity study in March, we are going to perform a first in vivo trials for these compounds. The third part of my talk involves PET MRI multimodal agents. I believe that were created within my group, and I believe that they are absolutely potential candidates to be used to one of the tissue engineering construct application. So we have synthesized iron oxide nanorods coated with silica for gallium-68. So we have incorporated onto silica of the nanorods the microcycles as a microcycle for gallium-68 coordination. This is the golden standard. We also added a PEG chain to improve the stability, and we also wanted to have a control without the microcycle. So we have the most stable, of course, stability in water was recorded for this one, which is obvious because of the pack chain. And as well, this sample was having the highest relaxivity. So we have performed a stability study after incorporation of gallium-68 in the human serum and apotransferrin for over 3 hours. And surprisingly, by that time, we could already see and concluded that there was no need for incorporation of the microcyclic ligand for successful gallium-68 incorporation. So we did a further analysis, but the conclusion out of that was that gallium-68 has been very strongly binded onto silica shell iron oxide, the surface coating, right? So we took the most stable and the highest performing sample with a coordinated radionuclide onto in vivo study. And maybe I can show you here video, because we wanted to be sure that gallium-68 can be stable also in vivo conditions. So we incorporated gallium citrate. We injected the radioactivity in the mouse to see where the radioactivity goes. So where we can expect the free gallium to be released from our iron oxide. So the highest uptake was observed in bladder. So okay, that was the primary information that we could obtain. So after functionalization of the nanoparticles and doing radiochemistry with it, we have injected into mouse model about 10 megabarrels activity, and we could, sorry for that, and we could see high uptake in the liver and no uptake in the bladder, which confirmed the high stability of this construct. These PET scans and the result of that were strongly correlated with MRI imaging that I've performed. As you can see, there was a strong T2 signal enhancement in the liver, and this correlates and is consistent with the PET data that we have obtained. So I will do that. So what I'm going to do in the close future is to assess the toxicity of the hydrogels and plan my in vivo experiments to assess the function of the hydrogels in vivo, and as well keep collaborating with Netherlands towards development of visible calcium phosphate cements. So as I said, a lot of tests are right now ongoing, and the first in vivo is already planned in March. And right now, I would like to acknowledge my supervisor, Steve Archibald, and Dr. Benjamin Bork and Juozas Domarkas. These guys in here for scientific support, and all my friends from the group, and of course sponsors, especially Marie Curie and King's College London. And thank you for your attention.